What's up? What's up? What do we got here? Oh, that's not good. Okay. All right. I need a sound check. I need video check. Let me know how the feed is. Let me know how the video is coming in. For those of you that are already here, I need sound check. I need video check. Please let me know in the chat how I'm feeding. All right, Jack. Glad you could be here. Liv, glad you're here since you need some help. We'll try to do that for you. This is crazy. I should be watching this NFL game on right now. You got the Rams versus the Saints. Saints are up by 2. The Rams just scored. Got me doing this live stream right now. Seriously, 41-yard touchdown pass to Cooper Kupp. Are you kidding me? Got Yasmin tripping in the next room. I don't know what's going on right now. What's happening here? All right. Yeah, LOL to you too, Jack. Please. This is not an LOL matter. I'm missing a very important game. Anyways, we'll wait for some people to pile in here. We only have like 5. I posted the link for the live stream on Google Classroom, so we'll see. I also made this public so people were notified if they subscribe to the channel. So we'll see if people start piling in here in just the next couple of minutes. So we'll get started shortly. Hang tight. Chapter 11, Genetics and Inheritance. Throw in Chapter 14 also. Let's see here. Yep, just 5 in here. All right, maybe that's all we're expecting right now. All right, so what I'll do to get things started is to first go through the textbook. I'll cover pretty much everything in order from sequence from section 1 to section 3. I'll skip section 4 of chapter 11 because that's on meiosis, and we pretty much already covered that pretty much 2 weeks ago. And then I'll get into chapter 14. We'll talk pedigrees and sex-linked disorders and recessive traits and all types of fantastic genetic goodness. All right, I think that's it. That's a few minutes in. Let's go ahead and get started then. Ava, glad you're here. Thanks for stopping by. I knew you would. You emailed me, so I knew it. Knew you'd be here. Jack, you emailed me too, man. Sorry about the I didn't post the what's it called? The C or no questions. I left it at work. It's sitting on my desk. So if you want to come in a little bit early tomorrow, maybe around 7:15, 7:30, you can take a look at that. As soon as I get in, I'll either post it or you can come and take a look at it. So my bad on that. But anyways, we'll cover everything today. So we'll get you all caught up. Riv, glad you could be here. Thanks for stopping by. Who else is in here? So we got Liv, Jack, Ava, Riv. Anybody else? It says 6, but oh well. We got some innocent bystanders in the place. All right, so I'm going to go ahead and get started here. Let me know if the sound cuts out or if it doesn't sound right. I'll try to get things adjusted. If the video feed starts getting off a little bit, let me know as well. All right. So what I'm going to do is I'm going to get started through the textbook. So I'm going to start with chapter 11, section 1. Okay, so what I have right now is I have the textbook right in front of me. I'm on page 308, looking at the work of Gregor Mendel. So all the P-plan experiments, all this background information. So you probably should know a little bit when it comes to the P-plan experiments. Make sure you're familiar with the process of fertilization. So, basically what he did in terms of crossing all the different types of P-plants, cross-fertilization, self-pollination, things like that. Make sure you're familiar with the difference between a pure breeding trait or true breeding versus a hybrid. So like, say, for example, you have the F1, which were all hybrid plants, and then the parental generation, the P, they were both pure breeding, but for opposite conditions of the trait. So I think that actually goes on to page 310. So it goes over all of the different terms. So it starts with what Mendel called factors, which ended up actually being genes. And the genes specifically were known as alleles, the different versions of the genes. So the principle of dominance, which Mendel essentially coined, he just basically took this idea of crossing the different types of plants for the different features, and he addressed how they interacted in terms of the offspring. So he's got seed shape, seed color, seed coat, pod shape, pod color, flower position, plant height. These were all examples of the characteristics that he analyzed. And the nice thing about these particular traits is they had two very distinct appearances or phenotypes. So for seed shape, for example, it was round and wrinkled, round being dominant, wrinkled being recessive. Plant height, it was tall being dominant to the short, which was recessive. So he went ahead and he did these crosses. And on page 311, you could see the 3 generations that he crossed. So it starts with the parental, pure breeding tall, crossed with a pure breeding short, and they produced all tall offspring. And then he did the same thing with the F1s. He self-pollinated them. It was, these 2 tall plants. And when he crossed them in a ratio of 3-to-1, it was 3 tall to 1 short. So figure 11-4, you could see the crosses and what he got. Now, obviously, at the time this was confusing to him. He expected to see that some of the F1s should have been small, but they weren't. He noticed that all of them were the tall variant. And of course, this was because the dominant allele was masking the recessive. So even though the parental generation had a short parent, none of the offsprings were short, but they did carry the allele, but it was being masked by the dominant allele. So this is the whole idea of, the principle of dominance and how it works and how, different traits are transmitted from one generation to the next, but sometimes they're covered by the dominant allele. So I believe on the test, there is some mentions of these crosses. So you want to make sure that you've gone over the parental, the F1, and the F2s. Besides the principle of dominance, also in Mendel's pea plant experiments, he established the law of segregation and how the alleles separate from each other during meiosis. That's essentially what this topic is. This has to do with gamete formation. We've talked about this in meiosis. That was section 4 of chapter 11 a couple weeks ago. So make sure you're familiar with that. And that pretty much covers all of section 1. It was basically just a history of the pea plant experiments from Mendel's side. Do keep in mind that Gregor Mendel himself actually was not a geneticist. He was, an Austrian monk, essentially, and he was a mathematician. He did a lot of work with probabilities and different mathematical equations to try to confirm his findings, but he himself was not a geneticist. But he did come up with a lot of the basic fundamentals of all the different topics in his research with the pea plants and how it inherited. They inherited their characteristics from their parents. So that's pretty much everything from section 1. Does anybody have any questions on section 1? Mendel's pea plant experiments, all of his findings from those things. That's pretty much it. All right, awesome. Kendall's here. Cool. All right. Abby, glad you could be here. Thanks for stopping by. If there's any questions on section 1, go ahead and ask now. If not, I'll just go ahead and proceed onto section 2. All right, so it looks like we're good. Let's go ahead and move over to section 2. Oh, okay. So Liv asked about the F1 and F2. So if you're going to understand F1 and F2 generation in Mendel's pea plant crosses, you have to understand the parental generation. So the parental generation were both pea plants that were true breeding. One was true breeding tall. The other was true breeding short. So if you think about it from the allele standpoint, the true breeding tall has 2 copies of the tall allele. So capital T, capital T. The short plant that was crossed with it was 2 lowercase t's. So if pure breeding tall crossed with pure breeding short, in the process of them producing offspring, they only produced heterozygous offspring. They were pea plants that had a copy of the tall allele and a copy of the recessive, the short. When you cross 2 heterozygous pea plants for a certain trait, that's the F1 generation. What you're going to get in your F2s is you're going to get a ratio of 3 tall to 1 short. You could do this with a Punnett square. So, if you got the capital T lowercase t crossed with the capital T lowercase t, you do the Punnett square, you'll see that you have a 1 in 4 probability of a pure breeding tall a 2 in 4 probability of heterozygous, and a 1 in 4 probability of lowercase t, the homozygous recessive, the short variant. So do the Punnett square, cross 2 heterozygous. That would be the F1s. When you cross them, you get the F2s. And you'll get the 3 tall to 1 short phenotypic ratio. That's essentially what it is. So the parental, you're crossing the pure breeding tall and the pure breeding short. You get all heterozygous tall. When you cross 2 heterozygous tall, you get that 3 to 1 ratio, 3 tall to 1 short. So do the Punnett square. You'll see the breakdown of all the allele frequencies and all of the different possible outcomes of the offspring. So that's pretty much it. You can get that particular cross. That's on page 311. You could see the parental, the F1, and then the F2s. All right, so moving on to section 2, this is applying Mendel's principles. Essentially, this is all of the probabilities in the Punnett square. So to reinforce all of Mendel's crosses. So this is essentially what he used in his pea plants experiments. These were the mathematical concepts that he used to reinforce it. All right, so I guess you got to start with what the alleles represent. So there are 2 alleles for every particular trait. So if we take the trait plant height, you have the tall allele, which is capital T, and then you have the short allele, which is lowercase t. The tall allele is the dominant phenotype or the dominant allele. So the short recessive, and each of them, of course, in combination creates a genotype. So if an organism has 2 of the same allele, we say it is homozygous for its genotype. If it has 2 different alleles, then we say it's heterozygous. Those are just some basic vocabulary terms for section 2. So there's pure breeding homozygous in the form of dominant, so 2 capital T's. Then there's homozygous recessive, which is 2 lowercase t's. So they say they're homozygous because they have 2 of the same allele. The heterozygous are also known as hybrids, so you have a capital and a lowercase allele combination. So those are essentially the same thing, heterozygous or hybrid. Now, genotype represents the allele frequencies, or the allele combinations, I should say. So capital T, lowercase t is a genotype. Lowercase t is a genotype. Capital T is a genotype. They are the letter combinations that represent the alleles. Phenotype is the appearance. It's how the trait manifests itself visually. So a phenotype would be, tall. A phenotype would be short. A phenotype would be, round seed or wrinkled seed coat. All right, those are appearances. They are visual manifestations of traits. So those are the phenotypes. The genotypes themselves are the allele combinations. Now, a gene is a section of DNA on a chromosome. At the time, he didn't know. Mendel had no idea he had no idea exactly what a gene was. He had no idea the existence of genes, but he knew there was something being inherited by the offspring from their parents. It turns out those are genes. Alleles are variations of genes. So the tall allele is the tall variant of a gene. The recessive allele is the short variation of the gene. So basically, genes, there's different types of genes, different variations. Those different variations are the alleles. So the tall allele, the short allele, the round, the wrinkled, all the alleles are just the variations of the characteristics. No, Riv, the gene itself, the genes are the expressive components of a trait. So a section on a chromosome is a gene, a specific section. That gene controls a specific trait. The traits manifest themselves, they're programmed and they're coded by the genes. So the different variations of the genes are called alleles. And of course, the genes express the proteins that express the traits. So that's kind of how you have to look at it. I don't want you to confuse it, but at its most basic fundamental understanding, the allele is a version of a gene, and it's the genes themselves that code for the proteins that express the traits. So if, certain genotypes are expressed, certain phenotypes are going to be visible. So in the case of, plant height, for example, the tall allele is going to code for tall plants, and the short allele is going to produce short plants. And that's pretty much how genes work. Whatever is dominant, whatever is recessive, it manifests itself based on the combinations of the genotypes. That's how it works. Hopefully that answers your question. If not, you can ask at the end. I'll definitely come back to it and try to re-explain. For those of you that remember doing all the different Punnett squares, we went over countless examples. We went over several, worksheets and exercises to reinforce the concept and the practice. So maybe go back and revisit some of those crosses and see if you can get those correct and check your work. And if you have any questions, you guys can ask those tomorrow before the test. One thing you want to make sure that you are able to do is make sure that you are able to do a dihybrid cross. There is one dihybrid cross on the test. A dihybrid cross is essentially the same thing as a monohybrid cross. You are crossing an individual with another individual for a certain characteristic or trait. That's a monohybrid cross. It's a single trait. So plant height, for example, crossing a pure breeding tall with a pure breeding short. That is a monohybrid cross. It's one characteristic or trait. A dihybrid cross is going to be two traits simultaneously. So that would be, round or wrinkled seeds and then tall or short for plant height. When you have those two characteristics, it becomes a dihybrid cross. Dihybrid crosses have 16 squares in its Punnett square. So there's a lot of different outcomes. There's a lot of different probabilities. You have to take the dihybrid cross in which you have 4 different alleles for each of the parents. So for example, if it's plant height and it's round seed coat or whatever. So if it's tall and round seed coat, pure breeding for both traits, capital T, capital R, capital R. So it's 4-letter combinations. You have 4 alleles. So make sure that when you're doing that, you identify each of the parents' genotypes correctly, and then you give all of the different combinations of all their possible gametes and all the alleles. So I would definitely look at page 317. Look at that dihybrid cross. I think the traits there, let me see here. Yeah, they go over round, seed coat, and also the color. And it's a good example. So if you want to go over those dihybrid crosses, they are present on page 317. We also did a couple in class. I know we had at least 1 or 2 that we did for bell work. So go back to those and make sure you can, allocate the alleles correctly so that you have all the gamete combinations for both parents, and make sure that you do that correct. All right, so that's it for section 2. Make sure you can do dihybrid crosses. Make sure you can do monohybrid crosses. Make sure you're familiar with probabilities. If I ask you for phenotypic ratios, or if I ask you for genotypic ratios, those are pretty simple and straightforward. If it's a monohybrid cross, it's got to add up to 4. If it's a dihybrid cross, it's got to add up to 16. If you guys have any questions on that, by all means, go ahead and ask. Or if you need to see an example, come and see me before school tomorrow. We can go over an example of a dihybrid or a monohybrid cross. Now, section 3 in the textbook has to do with other patterns of inheritance. So in this particular section, it kind of takes genetic inheritance patterns to the next level. Yeah, you know what? For Jack, I'll come back to independent assortment in a second. In terms of other patterns of inheritance, make sure you are good with not just complete dominance, which is what we saw with Mendel's P-plans. Make sure you're good with the incomplete dominance example. I gave you guys the example on page 319 with the 4 o'clock flowers. In this case, you have the red genotype and phenotype, which is 2 R's. And then you have the white phenotype, which is 2 W's. And when you cross those, you get a bunch of RW genotype combinations, and those are pink. Incomplete dominance always results in some kind of an intermediate phenotype, so something in between or blended. Which is different than codominance, where you have multiple alleles that are dominant and can coexist together. So incomplete dominance is more like the 4 o'clock flower example on page 319, and the codominance is more like human blood typing and calico cat fur color. I think that's pretty much it for section 3. I didn't really talk too much about anything else. The one thing I will say for the case of human blood typing, there's going to be at least 2 questions on the test. And I think both of them are actually written responses where you actually have to do some analysis, you have to do some crosses and some critical thinking. So the 2 questions that are on human blood typing, you actually have to make a decision. There's a bunch of criminals being accused, some suspects being accused of a crime, and they find certain alleles of blood types, on the crime scene. And they're trying to figure out who could have possibly have done it and who couldn't have done it based on the alleles of blood that they find. So make sure you understand that there are two genotypes for blood type A. You can have capital IA, which is pure breeding blood type A, and then you have capital IA, lowercase I, O, which is heterozygous. So you have a copy of the A, and then you have a copy of the O. That is still blood type A, but it has recessive alleles in the genotype. So make sure you understand that concept. The same thing goes for blood type B. You have the pure breeding B combination, and then you have the heterozygous with one copy of O and one copy of B. Blood type AB, which is a copy of A and B. And then to have blood type O, you need two copies of the recessive O allele. So make sure you guys understand that concept for codominance, with human blood typing being an example. So that's pretty much it from section 3. All right, so Jack, to answer your question, the law of independent assortment basically states that during gamete formation, during those meiosis events, you get a random assortment of genes that get assigned to each gamete. So keep in mind that in meiosis, you have 4 haploid gametes that are formed, which means of the 4 chromosomes involved in that split, 1 chromosome goes to the first cell, 1 form of the chromosome goes to the other cell, and so on and so on. So there's 4 chromosomes, 1 going to each version or to each of the new gametes. There's no rhyme or reason as to which one gets which chromosome. You just have to know that each one gets a chromosome. It's a completely random event. It's completely independent of each other. There's no rhyme or reason. It's just like the flip of a coin, or pulling the straw out of a hat. It just kind of happens. So if there's 4 different versions of a gene, each of the cells is going to get a different version of the gene. It's just kind of the way it works. So, when you think about independent assortment, just think of gametes forming in meiosis and how each one gets a different copy or chromosome from the splitting during segregation during independent assortment in meiosis 1 and also meiosis 2. So each reduction division during meiosis results in a separation of chromosomes and results in new cells, which they have new genetic combinations. Let's see here. Liv says, I don't understand what the I is supposed to be in blood typing. The letter I just represents human blood typing. That's all it is. So if it's a capital I, it's a dominant allele. So IA would be dominant A, right? IB is a dominant B. So a person with blood type AB has the dominant IA and the dominant IB. If a person has the lowercase I, that is the recessive O allele. So we write it as a lowercase I. It's as simple as that. So I'm not sure if they have it in the text. Let me see here. Yeah, I don't really see it in the textbook, but it is something that we went over in the notes. So yeah, that's all it is. Capital I represents dominant alleles, so it could be A or B. Lowercase I represents the recessive allele, which is blood type O. That's pretty much it. All right, so chapter 14 is on human heredity and human inheritance. So I think section 1 starts talking about the different types of human chromosomes. So it talks about the karyotype, which the karyotype for the humans, you have 46 chromosomes. That's our chromosome number. We have 23 pairs. So every cell in our body, with the exception of gametes, has 46 chromosomes, 23 pairs. The first 22 pairs, the first 44 chromosomes are autosomes. Those are chromosomes that are not sex chromosomes. The last pair, the sex chromosomes, in a female, those are XX, and in the male, they are XY. So that's pretty much what you have to know for that. Now, why this is important is because in certain characteristics and traits, some genetic disorders, some characteristics and traits are sex-linked, so they're carried on the X chromosome, which is one of the final chromosomes in the last pair. And then some are autosomal, and in that case, it's on chromosome pairs 1 through 22, chromosomes 1 through 44. So, and actually, Liv, page 394, the human blood typing is there. So it gives you all the different examples of all the different genotypes and alleles. Gives you the reason why human blood type AB is the universal acceptor and why blood type O is the universal donor. So for those of you that are interested in looking at that, that's on page 394. So that's pretty much it there. And then in terms of the sex-linked traits that I was alluding to, sex-linked traits are carried on the X chromosome, which means they're going to affect males and females a little bit differently. In the case of a sex-linked trait, males are often more greatly affected. So if you get a pedigree and males are the majority of those affected by a disorder, that is a sex-linked disorder. It is often where you have female carriers, but you can't have male carriers because they have the Y chromosome. More on that to come here with human pedigrees. All right, if you turn the page to page 396, you'll see a picture of a calico cat. I'll make this really, really simple. So when it comes to calico cats, this particular characteristics is a good example of a sex-linked trait. The standard situation with that is because it is a sex-linked trait, females are usually the only ones that could be considered calico. You need a copy of the black allele, you need a copy of the red or the orange allele. So, in the case of a sex-linked trait like this, males have XY chromosomes, so they only have one copy of X, which means they cannot be carriers for a trait. They cannot be calico cats. So in this situation, you have to be mindful. Males can't be carriers. They either have it or they don't. Females can be carriers. They can have it, they can not have it, or they can be carriers of it. So they're not affected, but they do carry the possibility of transmitting it to their offspring. So this is where things get complicated. So Liv, when you're looking at a pedigree chart and you're trying to decide if a trait is sex-linked, it's pretty simple. Look to see if you have a majority of individuals affected are males. So let's say that you're looking at a pedigree chart. There are 15 individuals in this pedigree, and there are 5 affected individuals. 4 of them are males. Then that is a sex-linked trait. If it's 4 affected males, 1 affected female, then there's probably a handful of carrier females, and there's most definitely going to be a sex-linked trait. So red-green color blindness, hemophilia, calico cat fur color, all of those are examples of sex-linked traits. In that case, female carriers, often you've got the situation where it's a 50-50 chance for all the male offspring they have. Half of them are going to have the recessive allele and half of them are going to have the dominant allele. So that's pretty much how it works. It's very obvious when you see a sex-linked trait. It's not something you're going to be wrestling with. If you see more males affected than females, like 4-1 or 5-1 or 6-2, that's most definitely a sex-linked trait. Now, if you look and it's 2 affected males, 1 affected female, or 2 affected females, 1 affected male, that's not a sex-linked trait. More than likely, it's not. That's probably not going to be the situation. Now, in terms of pedigree charts, there are going to be 2 pedigrees on your test, regardless of the form you get. So make sure you identify squares as males, females are the circles. If it's not colored in, it's not affected. If it's colored in, it's an affected person. If it's half colored in, that's a carrier. Remember, in sex-linked traits, only females can be carriers. You can't have male carriers of a sex-linked trait. Now, if it's not sex-linked and it's just autosomal, could be recessive, could be dominant, you can have carriers of a trait. Now, actually, autosomal dominant disorders cannot have carriers, right? You have it if you have 1 copy of it. Recessive, you can have carriers because you have 1 dominant allele and then you have 1 recessive allele. So be mindful of that. Some of the examples of some sex-linked traits we talked about was red-green color blindness, calico cat fur color, and hemophilia. Some autosomal recessive traits we talked about was like sickle cell anemia. I think that another one of the sex-linked traits we talked about was the balding gene, which is very common in males. That all comes from the maternal side. So, males often are frustrated with their balding and their receding hairline. That is something inherited from the maternal side. So, when a son says to his dad, "Thanks for making me bald," actually, that's completely false. It is the mom that passed on and transmitted the balding gene to her sons. Something interesting about the human sickle cell anemia trait, we talked about how some people are carriers. If you are 2 normal dominant copies of, the protein, the hemoglobin protein for blood oxygenation, that protein, which is, I don't know, the best way to explain it is sickle cell anemia is a recessive disorder, autosomal recessive. You need 2 copies of the recessive allele to express sickle cell anemia. You could be a carrier, 1 capital, 1 lowercase. That gives you the heterozygous advantage. the recessive allele makes you resistant to malaria. So if you have the heterozygous, not only do you not have sickle cell anemia, but you also have malaria resistance. So that is the heterozygous advantage. I think that's pretty much it. That covers everything. So if you guys have any general questions, my plan was to be on here for 30 minutes, help you guys review, give you guys a chance to ask any questions. If you have any questions on any of the topics we covered, if I overlooked something, if there's a specific trait you want to ask about or characteristic, any vocabulary that you're unsure of, go ahead and ask it now. I'll hang out for a couple of minutes and answer any questions you guys might have. I think I'm up to speed in the chat here. I don't see any unanswered questions. Now what I'm doing now is I'm just looking at the slide presentation that I posted to Google Classroom to see if maybe I, possibly skipped something. I'm not sure. Abby says, what was the autosomal dominant disease we talked about? That one was the von Willebrand disease. That was another blood disorder, I believe. Now, because, Abby, that one is autosomal dominant, in order to have von Willebrand disease, you just need one copy of the dominant allele. So if you're capital A, lowercase a, you will have von Willebrand disease. If you have two capital A's, you'll have von Willebrand disease. But in most cases, most people have the two lowercase a's. Von Willebrand disease is a pretty uncommon genetic disorder, but it is one that is autosomal, and it is also one that is dominant. So one copy of the dominant allele gives people von Willebrand disease. All right, so Kendall says, what's the difference between autosomal dominant and autosomal recessive? So autosomal just means it's not a sex-linked disorder or a sex-linked trait. The recessive part is the trait or the genetic disorder is carried on the recessive form of the gene. So sickle cell anemia is an autosomal recessive disorder. So to have sickle cell anemia, you need two lowercase a's. If you have a capital A and a lowercase a, you're just a carrier. If you have two uppercase a's, then normal shaped cells, normal blood oxygenation. So you need to have two recessive copies to have a recessive disorder. Regan says, do we need to know about Down syndrome and the unequal distribution of chromosomes? The only thing you really need to know for that, Regan, is just what non-disjunction is. So during meiosis, if you have an unequal distribution of chromosomes go to one particular cell. So say a cell has 4 chromosomes and it splits during meiosis 1, and one cell gets 3 chromosomes and then the other cell gets 1 chromosome. That would be an unequal distribution. That would be a non-disjunction. So that's pretty much all you have to know for that. Jack says codominance is where it's either one or the other. No. Codominance is when you have two alleles that are both dominant and exist together simultaneously. So human blood typing, blood type A and B, right? Blood type AB is codominant. All right, blood type A is not masking B. Blood type B is not masking A. They both exist simultaneously, creating a coexisting genotype and phenotype. That's how that works. Incomplete dominance is where you get kind of an intermediate mixed blend. It creates a different phenotype. So 4 o'clock flowers, you have a white variant crossed with a red variant, and it produces all offspring that are pink. So the incomplete dominance doesn't produce, the red or the white. It produces a pink. It's in between. It's a blend. It's a different phenotype. It's unique and intermediate. Codominance, they both exist simultaneously. So calico cats, codominance. Human blood typing, codominance. The roan coat of cattle, codominance. Incomplete dominance, we're talking about the 4 o'clock flowers where you're crossing the white and the red and you get pink. That would be the incomplete dominance. For those of you that have been looking at the slide presentation, some of the slides, they have an explanation of genotypes using letter P for flower plant, the flower color of the plants. You have the genotype examples are there. If you're looking for some review on probabilities, ratios, Punnett squares, monohybrid, dihybrid crosses, that's all there. You have different types of crosses, heterozygous, pure breeding, hybrids, phenotypic ratios, test crosses, it's all in there. Complete dominance, incomplete dominance, codominance, all of that is there. I recommend that you guys take a look, at least to kind of go through it. And then there's some links which can show you some examples. I recommend that as well. So definitely take a look at that. I forget when I announced this. Take a look here. It was the announcement October 31st. I posted that slide presentation. So there you go. That covers everything. Any other questions? I'm getting ready to kind of wrap things up here. Wanted to be on here for about 30 minutes, so it looks like I kind of hit my quota. And I'm out of water. All right, looks like that's all the questions. If you guys want, once this video posts, you can go ahead and ask questions in the comment section below. And I think I could also answer questions that way. If you want to just ask questions on the most recent announcement on Classroom, you guys can help each other, help you review. You can ask me. I'll try to chime in. You'll be able to reply and things like that. So if you have any questions, go ahead and post in the Classroom or just use the comment section below once the video posts. Ask your classmates, ask me, be supportive of each other. The test tomorrow, I think, is like 45 questions or so, 46 questions. I think it's about 40 multiple choice. There's probably 4 or 5 different genetic crosses and written response questions with explanations. It's pretty straightforward, pretty straightforward. I can't think of any trick questions on there. So hopefully that helps answers a lot of questions. Hopefully you guys feel prepared. You've had over a week to prepare for this exam. I announced this last Monday. So that's 6 and 7 days to review and prepare and ask your questions. So hopefully you guys do well. I think I'm going to leave it on that note. Again, if you have any questions, go ahead and do so in the comment section below on this video. Hit the slide presentation, read the textbook sections in the test, ask your friends, ask in the Google Classroom. We'll try to get to you as soon as we can. So that's it. I'm done. Long-winded, 38 minutes, longer than I wanted to be, but I hope you guys, benefited from the review. So that's it. Thank you guys for stopping by. Thank you for participating. Those in the live chat, thank you for your questions. I'm sure somebody found them helpful. And for those of you that are just innocent bystanders, thank you for being here and taking the time to try to improve your understanding on genetics and inheritance and reviewing for the test. So that's it. I'll see you guys tomorrow. Peace.